Damos nuevamente la bienvenida al Dr. Ken Stuart, presidente emérito fundador del Seattle Biomedical Research Institute. Dr. Stuart.
Buenos días. That is all my Spanish. May I ask those of you who speak English to raise your hands? All right, 20%, okay. First of all, it is an honor and a pleasure to be here today to celebrate the 50th anniversary of CIDE. I don't have enough time to express the joy that I have for Nancy and how honored I am to be here and how impressed I am with what's happened at CIDE since my first visit quite a few years ago in the previous laboratory facilities, which are very memorable. I also appreciate the simultaneous interpretation. This is something I could probably use at home at my laboratory and also in my personal home when I need to have converted what I say into what I mean. This is a very hard act to follow the previous presentation, and a new experience for me, but one I'll do my best at. This will be a little bit more technical, but still as heartfelt in my own way. So Nancy asked me to speak about research and innovation at the frontier of bioscience, so I'll do my best. And I will do this from an infectious disease perspective. So what is it that we're trying to do with infectious diseases? We're trying to understand the pathogen and the diseases that the pathogens cause so that we can develop ways to solve these disease problems and also to expand our understanding of all of biology. And the pathogens I like to think of in three forms in different orders of complexity. Viruses, which have very small genomes, but they use them very efficiently because they commandeer or hijack the capacities of the host and then cause disease by disruption of these host processes. And bacteria, bacterial pathogens, which have a few thousand genes, are very diverse. They can infect cells by getting inside or externally. They can produce toxins. They can do many different things. They are much more durable than the viruses and have many different ways of transmitting themselves. And then finally, the parasites, or the eukaryotic pathogens, which are much more complex organisms. They're the ones that I enjoy the most, I have to say. They have many thousands of genes. They're highly adapted to the host. And they have developed many ways to avoid elimination by the host's immune system. And sometimes it's the immune system that actually leads to the disease processes. So what are the challenges that one faces in trying to cope with these diseases and develop the solutions? Challenges that we're all working toward as we at CIDE and elsewhere. First, we of course have to select a strategy to address these goals, develop a new understanding and tools, and of course the word strategy has special meanings these days as we all do our strategic planning, so it's not only at the laboratory bench, but it's at the institutional level. And then I think probably more so today than in the past, we are very much aware of what the implementation will be of the findings that we make in the laboratory. And I think this is probably because we now have a greater ability to see the connection between the work that's going on in the laboratory and the implementation of that work in the clinic which is so well illustrated at CIDAIM, which has both the laboratory activity and the activity in the hospital. And I would add to this that the innovation, which is a difficult word to define in its detail actually, is not restricted to the laboratory, but it actually permeates many things that we do. And one should be looking to be innovative and productive at all stages of the endeavor of improving health. So the perspective that I'll give will be from infectious diseases and also from my own personal experience. I founded an institution that is a parallel universe to CIDAIM. It's the Seattle Biomedical Research Institute. It was founded 35 years ago in Seattle. This is a picture of our new building. A few years, this will be Nancy's new building. It will look something like this. These are wonderful laboratories that are located in Seattle. The climate is a little different than here in Columbia. It's enjoyable sometime in the year. We actually have a similar source of revenue, namely it's all competitive research grants. So we're what is called a soft money institution, and we have affiliated ourselves with a number of institutions to help us with our work. And so in this sense, it is very similar to CIDAIM. These are the characteristics of the institution, which you can read faster than I can say them. I would say that, and I'll tell you a bit about our activities, but we are now actually, we've become the largest private U.S. organization that's totally devoted to global infectious disease research. And to me, this is a stunning statement to make, given the magnitude of the problem of infectious diseases, to think that our relatively small institution is among the largest. It's a travesty, actually. So we work in these three general disease areas on viruses, bacteria, and the protozoan pathogens. As you'll see, our work is more directed in the viral area, HIV/AIDS, in the bacterial area, tuberculosis, and the protozoa, it's primarily focused on malaria and trypanosomatids, or the kinetoplastids, as you prefer to call them. And we have one large project where we're determining protein structures of many pathogens, and they just are listed to the side. So these are what we call the big three: HIV, TB, and malaria, and then other pathogens as well. And the approach that we've taken, and so this is a bit of the strategy, the approach, and trying to adopt the new technologies and the new approaches. So you see that we have focused on viruses, bacteria, and protozoa. They're pictured here, the different pathogens. But what we have done is we are now integrating the approach using systems biology, immunology, understanding of pathogen biology, and molecular cell biology and genetics in an integrated fashion to achieve the goals of develop basic understanding and develop solutions such as drugs, diagnostics, and vaccines. And as I'll talk about later today, we do not do this alone. We need organizations like CIDAIM and other organizations that we partner with to accomplish our work. And so the approach that we're taking uses complementary, integrated capabilities, which I'll go through in a little bit here. So we have diverse expertise which spans from pathogen biology through a lot of technical approaches, the understanding of molecular biology, biochemistry, etc., through computation and systems biology, which is another way of saying mechanisms to collect, store, interrogate, and interpret the ever-increasing amounts of data and information that are being accumulated by us and by others. Secondly, as many are doing now, we've had to increase the numbers and types of technologies that we are going to apply to the problem of global infectious diseases. And we offer facilities within which to conduct this research. And one of our more recent facilities is a challenge center where we have malaria-infected mosquitoes where we can immunize people and then challenge them with malaria and then interrogate the immune responses, et cetera. And I'll tell you a little bit about this in an example. And then, again, we're focused on these three types of pathogens. And then we use this approach, which is now called systems biology, which is an integrated approach, to now think about all of these and utilize all of the data that we're collecting to create a real new knowledge and a new understanding of these pathogens, the diseases that they cause, so that we and others can now develop new solutions to these diseases. So I will quickly go through the 4 programs. Because of time, I won't go into any detail. I'll leave my slides behind if you want to look at the details. And so HIV/AIDS, huge problem, probably the most challenging infectious diseases that scientists have confronted. We are focused on trying to develop broadly protective neutralizing antibodies and are using primarily an approach where we are presenting what are portions of the proteins of the envelope protein of the virus in structural ways to produce antibodies and then interrogate these antibodies as whether they would give us an idea of how to produce a vaccine where there would be antibodies that would prevent infection by many different strains of the virus. And we're also working to understand the immunological response, whether it's protection or disease, to the virus as a correlate of the production of a vaccine, but also to understand how the vaccine would work in the context of the immune response, either prior to or during infection. And there are 3 laboratories. I've listed the names of the people working in this area. The second area, it's actually our newest area, is tuberculosis, where 1 out of 3 people on the planet are infected with the TB bacterium. It's pretty stunning. They progressively, as time passes, have a greater chance of developing disease. We are focused on understanding the persistent stage of disease, and we're focused on developing drugs based on new understandings of this disease. And this is very important because, as you know, the drug treatment takes many, many months, current drug treatment, and many of the drugs, many of the TB bacteria seem to not respond to the drugs. And of course, there's drug resistance that's developing. The third area that we're focused on is malaria. You probably have seen the recent paper that the estimates of malaria are now controversial. It seems to be declining, at least in some places, or according to some people, but probably not as much as people had thought or at least hoped. We are primarily focused on developing vaccines for this drug, and I'll explain as an example what our approach is. And then the final area, and the area that I personally work in and know of Nancy's work in, are the trypanosomatids, African trypanosomes, leishmania, and Chagas disease. And our focus there is on the development of, primarily the development of drugs. There was a second part of my talk, but I think I won't present it because of time, but I can talk ad nauseum about the trypanosomatids if you're interested. So what is systems biology and how is this used to solve the problems of global infectious disease? It allows what I would call a holistic approach to describing the biological processes. When I began my studies as a student, this was called physiology. So physiology, which is but the difference between systems biology and physiology is in physiology you are looking at the organism, including humans, as a whole and understanding the phenomena and the processes. The difference is in systems biology now, you're developing knowledge about all of the component parts and how they function and how they interact. And so now you're beginning to understand the organism as a functioning entity, as a machine, as it were. And so the ability, the technological advances now allow a detailed understanding of organisms and disease as they function, and then to integrate this, and that's the important part of systems biology, to integrate this information so that you have an understanding at a holistic level. So it combines traditional biological approaches with high throughput tools, often called omics, the many omics, and it uses mathematics to understand the system, and then it brings these together. And one of the promises of systems biology is its ability to predict. This is very important. And the ability to predict allows more rapid progress, but also progress at a lower cost, both of which are very important. So the traditional approach to research, the reductionist approach, which was a huge advance, by the way, shouldn't be, we should recognize that the reductionist approach was a huge advance when it was first applied. It was the study, as we think of it now, of one gene making its RNA and protein and then looking at the function of that protein. However, as we all know, that there are tens of thousands of proteins in the human organism, also in some of the pathogens, and that these proteins are functionally interacting. So looking at one gene and its protein and the function of that protein is informative, but it doesn't really help you have the level of understanding at which the pathogens actually operate. They operate as in a more synergistic fashion. So what is the systems biology approach? So I've tried to diagram it here, where it's utilizing a variety of technologies. Genomics, where we have the information that's encoded in the genomes of humans or the pathogens, how these are expressed initially as RNA, as proteins, and then as the subsequent molecules, which are reflected in the metabolome and the other omics. And then this data is integrated using computation. It gives us an understanding of the interactions of these molecules, so the physical and functional interactions of the molecules, which are then leading to these properties that we could now talk about as a physiological process. So these are the component parts, how they actually physically interact and how they function. And then as one goes around this, it is an iterative process, a repetitive process. As you learn more about the components, what the functions are, ultimately you can derive solutions from this iterative process of understanding the organisms at the level of complexity at which they actually function. So this is the general approach of systems biology. So I want to talk a little bit about how this could be utilized. So how is systems biology utilized, for example, to develop a vaccine? So here's an example. So one can identify signatures of predictors of a vaccine, such as immunogenicity. So upon immunization with a vaccine product, one can look at protein responses, such as the immune system is producing, what the transcription is, the lymphocytes, and the types of cells that are responding to the immunization. And then use that to identify these networks of molecular interactions that are leading to either the result in immune protection or not immune protection. And then one finds out what these networks, functional networks, so these are all the steps and activities that are occurring in response to a vaccine. One sees these steps, one can see what will work and what will not work. Then one can adjust the vaccine, perhaps add an adjuvant, see how that actually helps or doesn't help, and then go through this iterative process and then continue to refine what is happening to try to improve a vaccine. So I want to give you, well, I'll just give you one example. I won't get to the second example because of time. So I'll show you the approach that we're taking to malaria vaccines. So the approach that we're using employs a combination of computation and genomics to identify parasite gene function as it's occurring at the liver stage. So this is the strategy that I was talking about early on. So we have to make a decision what we're going to do. So we're focused on the liver stage because when the malaria parasite is introduced by the mosquito, the first thing it does is it goes into the liver and infects the liver cells. It then reproduces in the liver cells, but at that stage it does not cause disease. And it's been demonstrated initially in mouse model systems, but then ultimately in humans, that if you disable, do not kill, but disable the parasite so that it cannot complete the liver stage of its life cycle, but it dies, but it lives for a while. Then, when you try to reinfect a person after some time when they've experienced this, they are protected from infection. So this was initially done with radiation-attenuated parasites. So this is the equivalent of an attenuated vaccine, although it's not a product. It's not able to be mass-produced as yet. So this is the demonstration that there can be sterile immunity to genetically disabled parasites. So what has been done primarily by Stefan Kappa's lab at our institute is to identify which genes are utilized by the parasite to complete the liver stage of the life cycle, and then to genetically engineer the parasites and remove those genes which are essential for that completion of the stage of the life cycle. Fortunately, those parasites can still be grown in culture in the blood stage. They can still transit through the mosquito, and then they can be delivered currently by mosquito, but maybe in the future by a syringe, into people, and the prediction is that they will be protected much, in fact, more effectively than using radiation-attenuated parasites because they will all be alive. They'll all be homogeneous. They'll all be the same, whereas there's a range of characteristics of the radiation-attenuated parasites. We actually initiated a first clinical trial in collaboration with Walter Reed Army Institute for research, and the showed that the parasite is attenuated. However, some of the parasites were able to transit, of this first variant, were able to transit the liver stage and get into the blood stage and result in an infection. So that means that we've learned something, but not enough. So that's one turn of the cycle. And now what we're doing is we're analyzing the blood, especially the immune responses to these parasites, to try to understand what would be the basis of protection. So out of 6 volunteers, 1 got infected, 5 did not. We had to stop the study when that one person got infected. So this is an example of the approach by which systems biology can be used to develop a vaccine. Also, I would say to learn more about the parasite, because we know what these proteins are. They are proteins the parasite makes and puts on the parasitophorous vacuole, the membrane-bound vacuole that the parasite lives in within the liver cell. We know that at least one of these proteins is involved in bringing fatty acids into the parasite, which it probably needs to reproduce, probably partially disable the parasite, but did not completely prevent the parasite from completing that stage of the life cycle. So we learned a little bit more about the parasite and need to go back to make other variants of this vaccine. So this is a diagram of this approach and how we're extending this. So this is I'm not showing you the complete life cycle of malaria, so that's something different. Usually a parasitologist will get up and give you the life cycle, but what I'm doing is I'm focusing on the liver. So in the parasite, which initially comes in, it's called a sporozoite, looks like this. If it's radiation attenuated, it can infect the liver cells, but it does not multiply very much. So it doesn't multiply enough to get out of the liver. We have made mutations, and I say we, it's primarily Stefan Kappa's lab, where we've removed genes, different genes. So in some cases it can develop a little bit in the liver. In other cases, if we remove these two genes, it can develop a little further in the liver. And the wild type can go all the way through the liver and then make these merozoites, which burst out of the liver and then infect the red blood cells. So what we're doing now is we're developing genetically attenuated parasites which will arrest at this later stage of the liver stage development to present as many antigens as possible. And we're also using other techniques to experimentally interrogate the immune response that is leading to protection by stopping the development of the parasite using drugs, anti-malarial drugs. In this case, primaquine, where we will infect people. In fact, this is a clinical trial that is running right now at our institute, where we're infecting volunteers with malaria, then we're treating them with primaquine, which will stop their infection at the liver stage. There are studies that we're also doing and others are doing where we're blocking the blood stage of infection using another drug, chloroquine. Then we're interrogating the immune system and using that to understand both the immune response and also to identify the actual proteins from the parasite which are responsible for protection, so that ultimately the vaccine will not be a live parasite, which is probably actually the best vaccine, but it's too difficult to produce and deliver, but to identify the protein components that are responsible for the protection, so there can be vaccines which are more easily and inexpensively manufactured, more stable, and able to be produced and then delivered in the field. So this is a diagram of the systems biology approach. These are actual experiments. I won't go through the details unless you're interested, where we are working now to identify the antigens, both that could be vaccine candidates or genes that, for these antigens that we will eliminate to make genetically attenuated parasites. So this is a vaccine that's in the field now, this RTSS. This is based on one of the proteins. Many years have been devoted to this effort, so we're involved in looking at the immune response to this vaccine. These are genetically attenuated parasites. And then this is a picture of liver cells from mice in a mouse model system where we've labeled the parasites with a fluorescent label so that we can take individual liver cells from dissociated livers and then using flow cytometry collect these and then identify the proteins in these liver cells, the parasite proteins that are being expressed in this liver stage. So these are to identify the antigen that can become part of the vaccine. Then we are looking at the immune responses. This is leading to this network of identifying the proteins that are involved in the immune response and how they are functionally interacting. So this is that network, the networks that we're talking about. And then this is an example of a clinical trial to show that we'll then do the clinical trial and then we'll go through this iterative process and then apply these analyses which are to understand the networks of responses in the host, what are the correlates of immunity, what that immunity is directed against, namely these antigens, and then ultimately to identify adjuvants and to formulate vaccines. So this will be the direction that we will go in the future. So this is an example of how one might use systems biology once you've identified a protective antigen, how you might optimize that antigen. So the RTSS story is a good example because the RTSS vaccine was based on a circumsporozoite protein. However, it turned out not to be very immunogenic. However, with an adjuvant, the adjuvant now has made that into a vaccine that has efficacy. So this is an example of how systems biology would interrogate the response to a vaccine with a particular formulation. And then a second formulation, which could be a different adjuvant, for example, that would lead to a different network of responses. and then based on that, one would choose which would be the better antigen, better adjuvant, and then use that process to test different adjuvants. So I was going to talk about the trypanosomatid pathogens, but I don't have that much time. But I'll just go through this very superficially so you'll at least have a flavor. So we're focused on 3 trypanosomatid pathogens depicted here. You may be familiar with these diseases. The important thing is that they have related genomes. So these are the genomes of 3 pathogens that are aligned. So this is the Trypanosoma brucei, Leishmania, and then Trypanosoma cruzi here. These genomes are very closely related. They have very characteristic differences, and so circled here are the surface antigen genes of trypanosomes then that are characteristic and used by the parasite to evade the immune system. So it's what's similar and what's different is really very useful here. So using proteomic approaches, we've been able to identify proteins. We've actually now, by mass spectrometry, been able to see half of the proteins predicted by those genomes, which is just a prediction of what are the proteins that are produced by the genome. We've been able to improve the annotation of that genome, identify what proteins are functioning and what pathways in the processes, the metabolism of trypanosomes and Leishmania, since they're very related, which is very important for identifying what would be drug targets. And then to use a variety of methods, I'll just go through this. We focused initially on the mitochondrion for a variety of reasons, identified all of the proteins in the mitochondrion of these organisms, which there are 1,100 proteins in the mitochondrion, most of which are present in multi-protein complexes, which is very useful because half of the proteins that are predicted in the genome have no known function. They're hypothetical proteins. But by associating them physically with proteins with known functions, we know what the general functions now are of these proteins. So it really helps us understand the functions of these proteins. And so I'll just run through this. We can develop networks for these complexes, which are showing the physical interactions of the proteins, which helps us understand the functions. We can see, for example, the ATP synthase, that the physical interactions that have been identified by just looking at mass spectrometry using isolating these the ATP synthase complex by virtue of each of the individual proteins. This now fits exactly the crystal structure of the protein, so we know that it's validated. So it helps us understand the novel proteins which these parasites have that differ from the host, and that's important. That make them potential drug targets. I'll just run through these. And so using this, we've been able to identify what are potential drug candidates and then identify that we can look at the mitochondrion as it's integrated within the whole organism. So this is an example. Here's the nuclear genome, which encodes roughly 10,000 proteins, 1,100 of which go into the mitochondrion. So these are the general functions of the mitochondrial proteins, some of which encode the respiratory system, the energy generating system. And then the mitochondrial genome makes 18 proteins. Those 18 proteins translated in the mitochondrion. So there's another cell within the cell almost. These make some of the components of the oxidative phosphorylation system. So by using this approach, we can now get a more comprehensive understanding of how this organism is developing some essential systems such as the oxidative phosphorylation system, components of which are now candidates for drug development. So in conclusion, I know I ran through this last part rather quickly, but it'd be nice to get back on track. The systems biology approach is an integrated application of multiple technologies. It's a powerful way to illuminate critical aspects of the pathogen biology and how it's interacting with the host, and namely how the host is responding to the infection, to allow us to develop new tools, and these are desperately needed, to combat these infectious diseases. This power, which is a technological power, is further enhanced when it's combined with innovative and thoughtful approaches, some of which are institutional, some of which are within the laboratory, to seek these solutions to infectious diseases. And the innovation that's applied actually operates at all levels, not only at the technical level, but at the institutional level and the individual level in the laboratory. And even in the choice of how you would design a single experiment to study an important aspect of a global infectious disease. So there are many people, too many people that I can attempt to thank for the information that I've presented to you. These are the people whose work that I actually showed some experimental evidence for. And I would like to, again, congratulate Nancy and all of the others who have worked so hard and so effectively at Sabine to make it such a successful story. Thank you very much. If there are any questions, I'd be happy to answer any questions.
Dr. Stuart, to congratulate you on your lecture on Efraín Otero. Yeah. It's a very nice presentation you do in your research institute, which should be a model for all of us. Could you, in detail, tell us how do you balance the problem between the budgeting for research grants and the overhead costs, which is one of the main problems we suffer here.
Yes.
Thank you.
So the question is how to balance the cost of doing the research with the real cost of having an institution where the research can be done effectively. And it's interesting you would ask that question, because one of the challenges is to explain to the investigators the importance of the cost of maintaining the institutional infrastructure. And so I have said many times that there's only one goal in for the administration of an institution, and that is to create the research environment where each investigator can be most successful. That's the only thing that we're trying to do. And everyone, including everyone, no matter what their role in an institution is, has to work toward that. This is a huge challenge. So we have a fundraising effort. We need to raise funds outside of our research project funds to sustain the institution. We have a board of trustees who help us with this, not as much as we would like, because our board of trustees are not wealthy people. So we reach out to the community and to foundations to get these resources. And I guess since the topic here is innovation, we also have to be financially innovative. So when we work closely with the investigators when they are submitting a grant, to have them include in the grant to the extent possible what are going to be costs to help sustain the institution as well as their project. This is actually quite difficult because every one of us realize that the cost that supports the institute is cost that comes from the research. But actually the two are intertwined. So we cannot do the research if we don't have the institutional facilities. We do not have I would say, a long-term solution to this problem yet. We do not have an endowment. We do not have intellectual property that is bringing in revenue. We wish we did. We only have our own intellect and energy and our ability to convince others to support us in our mission. And I think the surprising thing to us is how little the public is aware of the problems of infectious diseases. Astounding. And we think that we are telling many, many people about these problems. And we find that it's an extremely small fraction of people who have any reasonable awareness of the problem. But when they become aware, they immediately want to support. So I would say that for the long term, the most important thing is advocacy. To be able to reach out to the general public, politicians, business leaders, members of the community and say, this is your problem. It's actually one of the most important problems of humanity, and you need to help support it. That's what I would say.
¿Hay alguna otra pregunta? Just, I have a small question.
Yes.
So, systems biology, I think by definition, is a collaborative effort. What type of collaborations do you think are important in order to have, for example, in your case, this endeavor to become a success story?
So there's basically two types of collaborations that I think are needed, and I'll just speak to the technical side of that. One type of collaboration is within an institution, within a laboratory, between laboratories. There is a need to share knowledge, ability, space, technology. That needs to be done. Secondly, and I'll talk about this in my second presentation a bit, no one institution, and I would say no one institution, has in-house the ability to do all of the studies that are needed to really solve a problem. And so one needs to reach out globally, in fact, to get the complementary, and that's important, complementary, the complementary capabilities to achieve common goal. So we have a wide-ranging network of collaborations to do this, and including we'll be talking on Wednesday about possibility of developing other collaborations so that we can achieve the common goal. This again is a challenge to identify the best collaborators. There is no mechanism, no organized mechanism to help us do this. There is no LinkedIn, if you use that, I don't, but or any Facebook mechanism to help identify the best collaborators. Many years ago, I naively approached WHO, probably TDR, I don't think Carlos was, this was before Carlos' time, to try to identify some collaborators in the developing world to help me. And I would say the situation hasn't changed that much. So at some point, there are some things that have changed. There are these enterprises that have sprung up and these alliances that are working toward this. But this is another huge challenging area. So I would say that the efficiency with which biomedical research on infectious disease is moving, the efficiency is at a very low level because we haven't integrated the research system toward the common goals that we have. is a very chaotic system. So one of the challenges, and this may address some of these infrastructure issues, we don't want to have redundant infrastructure. We want to utilize the infrastructure that's available. I hope that addresses your question.
Thank you. ¿Alguna otra pregunta? Bueno. Bueno, gracias. Thank you very much, Dr.
All right.
Stuart.
Thank you. Thank you.